Hi, welcome to yet another session of biology classes with Lakshmi Chandra. Hope you have subscribed the channel. If you have not, please subscribe the channel and don't forget to press on the bell icon so that you get the notifications whenever I upload videos. So far we have been dealing with molecular basis of inheritance, different topics under that. And we have taken a very important molecule, that is DNA. I have explained the features of DNA and how this DNA was discovered to be the genetic material. So all those things are over. Now let's see how one DNA is going to replicate into two. How one DNA forms another DNA. Okay, how one DNA is going to replicate. So in that case, I hope you might remember the cell division that you have learned in standard 11, cell division. So in that cell division, what is happening is one cell is dividing into two. So you say ultimately the cell has multiplied. So in biology, whenever a thing is dividing into two, the net effect is what? Multiplication in number. So same process happens here also in DNA replication. It is DNA getting multiplied. Okay, so let's see how DNA is getting multiplied in a step-by-step process. So first, in this topic, DNA replication, I'll try to make you understand the overall mechanism of DNA replication. Okay, and its whole process of DNA replication is done by different enzymes. So enzymes are the key players in DNA replication. So what you have to know in this topic is what an enzyme is doing in DNA replication. So if you understand the role of different enzymes in this process, DNA replication will be very clear to you. So let's understand the overall process first. So here, in this process, the first and foremost thing that you should know is DNA is placed inside a cell. And inside the cell, in the case of eukaryotes, it is placed inside the nucleus. If it is prokaryote, it is placed in a space called as nucleoid. So, this DNA which is there inside the cell, inside the nucleus, it is placed where? It is placed as a conglomerated mass in chromosomes. Okay. And when does this DNA replication take place? It takes place during the S phase of cell division. During the S phase of cell division, DNA replicates. So when this cell is going to divide, cell undergoes different stages, G1, S, G2 phases. So it is during that S phase or synthesis phase that new DNA molecules are getting synthesized. So first and foremost thing that you should know is during which phase this is happening. During S phase of cell division this is happening. So DNA replication is part of cell division. Okay, part of cell division. So here, as you can see from this diagram itself, this DNA molecule is present on the chromosome as a conglomerated mass, like Maggi noodles it is there. So first what happens is these chromosomes become distinct. They become distinct. Distinct in the sense they acquire their structure. Like this, the chromosomes acquire their structure. They will have the arms and you can clearly see the constriction or the centromere. So first chromosomes appear like this. And on this chromosome, we have learnt last class packaging of DNA where I have told you how DNA is packaged in a chromosome. Okay, with histone octamer. So on this chromosome, our DNA molecule is present as a double helix. So this DNA which is present as a double helix, on the chromosome, this has to replicate into two. So first and foremost thing is for this DNA to replicate into two, its two strands have to be separated. These two strands have to be separated. It has to be opened up. The two strands have to be opened up. Okay. And when these two strands are opened up, so one strand is here, another strand is here, these two strands are opened up. How can I distinguish between these two strands? Based on their polarity. So one strand is running from 5' to 3' direction. Another strand will be running from 3' to 5' direction. So these two strands first have to be separated. These two strands are separated. But it's not very easy to separate the two strands. One cannot just pull it up and separate the two strands. So what has to be done to separate the two strands? We have to break the bond that is uniting the two strands. So now I think it is clear to you. And which is the bond that is uniting the two strands? Exactly, it is the hydrogen bond. So first and foremost thing in DNA replication is to remove this hydrogen bond which is actually holding these two strands together. So when this hydrogen bond is removed, the two strands get separated. Fine, the two strands get separated. These two strands are separated because hydrogen bond is removed. It is no more bonded with each other. The two strands are separated. So in this separated strand, if it was adenine here, in this strand it would have been thymine because of complementary base pairing. And if it is guanine here, it should be cytosine on the next strand, isn't it? Again, if it is guanine here, it is going to be cytosine. If it is thymine, it is adenine. If it is cytosine here, it is guanine, likewise. So based on the complementary base pair rule, the bases have now got separated. The hydrogen bond is now separated. Now there comes the role of the enzyme. Okay, so first enzyme here involved in DNA replication is helicases. Helicases. See, from the word itself we can understand. Helicases. It is unwinding the helix. How? By breaking the hydrogen bond. So, what is the role of helicases? Unwinding the helix. Unwinding the helix. How? By breaking the hydrogen bonds. Now the two strands got separated. Now when Watson and Crick has proposed the double helical structure of DNA, at that time itself they had predicted that DNA replication will be based on the complementarity of the two strands. And that was very, very true also. Okay. Now when these two strands are getting separated, new DNA strands will be formed which will be complementary to the existing strand. So here if a new strand is formed, that new strand will have bases that is complementary to the existing strand. If it is C here, it is G. If it is A here, it is T. If it is C here, it is going to be G. So this strand which I have written now, this is the new strand that is formed. Now what happens? These two strands will make hydrogen bonds with each other. Similarly, the other strand which got separated, that strand will also form a new strand. So if it is adenine, which is going to be thymine, guanine, cytosine, likewise. Okay. So this keeps on happening. So this strand which is running in 3' to 5', that also had synthesized a new strand. Based on what? Based on complementarity. So ultimately what you got here? You got this is one DNA molecule and this is another DNA molecule. So this DNA molecule has replicated into two different DNA molecules. Okay, so when I say this, you will feel, okay, this replication process is so simple, but it's not so. Here I have just told you the overall mechanism. Now let us take one by one, and I've told you all these steps are done by different enzymes, but I had introduced only one enzyme to you. Let me introduce another enzymes also. So here, overall process is this. So the first step is what? Unwinding of the helix. Unwinding of the helix. Okay. Imagine two copper coils two copper wires are there and you have coiled it like this in a helical manner. Okay, and you are trying to pull from between. And immediately if you just release your hands, what will happen? These coils can again recoil back. Same thing happens in our DNA also. So, as I have told you, in the first step that is unwinding of the DNA helix is done by the enzyme helicases, but the process or the effect that the helicases has done will go futile if the DNA strands are not able to keep them separated. So there are some forces which keeps the unwound DNA strands separated. So let us look into those forces. Okay, or those enzymes. So the second enzyme, Topoisomerases. Topoisomerases. Second enzyme is coming into action. This enzyme is trying to keep the two strands separated. Okay. So this enzyme is also called by another name called as nick and seal enzyme. Nick means cut and then seal. What is this cutting and sealing? Let us see. So for that, let me just draw this double helical DNA in a better manner. This is double helical DNA. Okay. Now imagine the helicase enzyme is acting here and it is going to break this hydrogen bond. You should remember one thing. Never this helicase enzyme will go and unzip the whole DNA together. It will never do. As I've told you, the two strands are fully separated and new strands are getting formed. Never two strands of DNA are fully separated. As in how the strands are getting separated, new strands will be formed. Okay, as the two parental strands are getting separated, new strands will be keeping on forming. And ultimately the two strands will be separated. So when the two strands are fully separated, what is happening is double helical DNA, daughter DNAs are now fully formed. So the process of separation and the process of formation of the new strands takes place together. Okay, takes place together. So first let us take a portion here. Imagine this is the area where DNA replication has begun. See, why I have taken this area is because, see, DNA strand, as we have learned, it is a very long process, a very long strand. It is approximately 2 meter long DNA. And if an enzyme is going to start the process from here, DNA replication process from here, it will take a long time to reach the other end. To complete this process, it will take a very long time. So, another option is that one enzyme can start from this end, another enzyme can start from this end. Again, it will take a long time because it is just one part of cell division. This process is taking place. Still, there are more processes to be completed for the cell to divide. So, if this DNA replication is taking a lot of time, it will lag the process of cell division. So, DNA replication has to take place at a very faster rate. And for that, DNA replication doesn't begin from one end or the other end, but it happens at different positions in a eukaryotic DNA. It happens at different positions in eukaryotic DNA. Okay. For example, I'll tell you, see, if the DNA replication is beginning, I'm just drawing DNA as two parallel lines, okay? Instead of like this here, I'm just representing it as two parallel lines and there is hydrogen bonds between them. Imagine DNA replication has begun here, one side, this is another side, this is third side, likewise, okay? This strand is 5 prime and so this will be 3 prime. So this is again going to be 5 prime and 3 prime. Okay, the two strands are like this and DNA replication is beginning at different areas. So the areas where DNA replication begins you call that site as origin of replication. So there will be lots and lots of origins of replication in a eukaryotic chromosome or in a eukaryotic DNA, because I told you it has to be completed very fast. So in lots of places this is being done. So for example, you imagine you're going to build a wall. You're going to build a wall, okay, and you know it has to be built from point A to point B. So, and from this point to point B, you have to build a wall. So if you keep just one person to build the wall, he will start from point A and he will keeping on building to point B. If you keep two persons to build the same thing, A will build and B will build. So both of them will build from both the ends and ultimately they will join the wall. So if you recruit some 10 people, what will happen? Say one person is here, second person is here, third person here, fourth here, fifth here, likewise. So this first person here will start building the wall this side. Another person from here will start building the wall the other side. So this person will start to this side, likewise. So if you recruit more and more people, the building of the wall will be done at a faster rate. Same thing is happening here. At different points, the DNA replication begins. Okay, from different points the DNA replication begins, and you call each and every point where it begins as origin of replication. And always from this origin of replication can happen in both directions, towards this direction and also towards the other direction. Towards both directions replication can happen. From here also origin of replication, if this is another origin of replication, if I put it as ORI, origin of replication, it can go towards this side and also towards this side. Okay, same way here also origin of replication can be this side and also towards this side. So this you have to know first, from which direction to which direction. So wherever is this origin of replication, at that point what happens? First the helicase enzyme will come and it will break open the hydrogen bonds. So imagine if this is the origin of replication and here the hydrogen bonds are broken. If the hydrogen bonds are broken, there what will happen? This is like this, hydrogen bonds are broken. So, and in that area the strand got separated. In this area the strands got separated because hydrogen bond is broken. So what you can see here? You can see here a big bubble. A big bubble. This big bubble you call it as the replication bubble. The replication bubble. So when does this happen? At the origin of replication, when the helicase enzyme is coming and it is breaking the hydrogen bonds, at that time the two strands they are getting separated. So you are getting a replication bubble. So if here this is another origin of replication, imagine, then here at the same area here also you will get another bubble. Okay, so this is origin of replication here and hydrogen bonds are getting broken, you got a bubble. Now for understanding, I am just dividing this into two, because I told you if this is the origin of replication, from here replication can proceed on either direction, So from which direction to which direction replication is proceeding? It is always from the open end of the bubble towards the tail part, towards the closed end it is proceeding. Always it is like that. That means always from the open end. This is the open end of the bubble towards the closed end. That is from here to this direction. And what about the other side? Open end of the bubble is here towards the other direction, closed end. So always it is like that. Wherever you can see this hydrogen bond is getting broken, in all those places bubbles are getting formed, and always the replication direction will be from the open end of the bubble towards the closed part. It is like that. Okay. So here this is the direction. Now imagine another bubble is getting formed here. Another bubble is getting formed here. Let me draw there for you to understand it in a better way. Another bubble is formed here. How that is formed there? Because here also helicases has acted and it has broken the hydrogen bond. So another bubble is getting formed there. So these two here, these two strands are now coiled. But here it is coming, another bubble is getting formed. Another bubble is getting formed. Okay. So here also, this is the origin, exact origin. If you divide this like this is the exact origin. From here it will close, it will move to the area where the end is closed. So one is proceeding from this direction to the other end. From here also, the proceeding in the opposite direction. So like here, one is proceeding towards one direction, from the other side it is proceeding towards the opposite direction. So as it is proceeding from one end to the other end, new DNA strands are getting formed, and ultimately what will happen is these two bubbles, this first one and the second one, will form a big bubble like this. It will form a big bubble like this. Okay, as it is proceeding, these here also hydrogen bonds will be broken and this will form a big bubble, and as this bubble is getting bigger and bigger, new DNA strand will be formed. New DNA strand will be formed, that is according to the complementarity. Okay, according to the complementarity, new DNA strand is getting formed, and as it is proceeding towards the two ends, from the open end towards the closed end, this is always proceeding. Ultimately, you will get two new DNA molecules, as I have told you. So, remember, never these two strands are completely separated and then a new strand is getting formed. It never happens like that. As and how these strands are getting separated, at the same time, new strands are getting formed. Now, let's go back. We were here talking about topoisomerase, nick and seal, I was telling you. Okay, they are helping in keeping the two strands separated. So let us see the role of topoisomerase or nick and seal enzyme, topoisomerase. So here, as I have told you, helicases is coming and unwinding the helix. You got a bubble called as replication bubble, but then I told you it is having the tendency to rejoin back. You have pulled it down, it will have the tendency to rejoin back because there is a coiling tension. Okay, now what is this topoisomerase doing is, it will come and it will make a cut here, say in these two areas. It is making a cut here. And when that area is cut here, when this is cut here, what happens? The coiled part, it gets uncoiled there. You imagine you're taking a rubber band and you're twisting from the two ends. So you can see their wave-like coiling getting formed. Okay, then what will happen if you just cut any portion in between that, the coiling gets recoiled immediately because the tension is getting relieved. Same way, in our DNA also, it was coiled and kept like this here. It was coiled and kept like this here. Only thing is the bond between them is broken and so the strands are getting separated. But still the coiling tension is there with it. So that coiling tension is removed by a cut. It is cut, and then when it is cut, what happens? It twists back to the original position. Tension is relieved. Okay, and then after that it is getting sealed back. So again there will be another cut. Tension is relieved, it will be sealed back. Likewise, it happens in almost all regions of this particular bubble. So in different areas of the bubble, topoisomerase will go and will first cut certain regions, then it is twisted back to relieve the tension, and then it is sealed. So what will happen? Even if you remove this force, that is, even if this bubble is getting separated like this, that force is removed, it will not immediately recoil back. Okay, so that is how topoisomerase is preventing the recoiling. It is making a cut and again retwisting it and then sealing it, rewinding and then sealing. So that tension is relieved. So this is actually relieving the tension of the helix. So releases the tension. So you call it as the torsion or the torque. Okay, we can write in simple term, releases the tension in the strands. It releases the tension in the strands by nicking, that is cutting and then recoiling. But then again you can see these are hydrogen bonds which were actually holding these two strands together. And those hydrogen bonds are now broken by helicases. And you know, hydrogen bonds are weak attractive forces, and when more and more hydrogen bonds come together, they become stronger. Okay, so here there are lots of bases present, and naturally there will be lots of hydrogen bonds present. So these hydrogen bonds are strong enough to come back and coil back. So we have to prevent again it from coming back and coiling. For example, you take two magnets, one is North Pole and another one is South Pole. Okay, if you just release your hands, what will happen? Immediately they will come and attract together, isn't it? So there should be some clips on either sides to hold these two separated strands. So which are those clips which is holding the two strands, keeping it separated? So that is the next protein here. They are called as single strand binding proteins, or SSBPs. Single strand binding proteins. What do? They come and bind to these single strands at different areas. They act like a clip. So this is a single strand binding protein here. Here you have another one. Here you have another one. Here another one. Likewise. So these single strand binding proteins will come and hold the two strands of the bubble. It will keep it separated until and unless the process of replication is getting over. So we have learned about how the two strands are getting separated. For that, first and the foremost role is played by the enzyme helicases. It comes and breaks the hydrogen bonds. So the two helix are getting separated. It will form a bubble-like structure. Now the next process is to keep the two strands in that bubble separated. Okay, that is done by the enzyme Topoisomerases, which releases the tension of coiling, and also by single strand binding proteins, which comes and binds to these single strands and keep them separated. Okay. Now, then hereafter comes the main enzyme for replication, the key player of replication. Okay, the main enzyme that is involved in replication, it is called as DNA polymerase. So, main enzyme involved in replication, it is DNA polymerase. DNA polymerase, because it is polymerizing the DNA, so the name itself is DNA polymerase, or it is also called as DNA dependent DNA polymerase, because depending upon the DNA strand, it is synthesizing new and new strand. So it is called as DNA dependent DNA polymerase, or simply DNA polymerase, or in short we also write it as DNA Pol, P-O-L, DNA polymerase. So this is a key player in DNA replication. Okay, this is a key player in DNA replication. Now we should know certain features of this key player. That is very important. Certain features of this key player. What are those key features? First feature, it cannot initiate replication. It cannot start replication. So first feature is, though this is a key player, it cannot initiate replication. It cannot start. It's like, imagine a train. It cannot add the engine, but it can add the bogies. One after the other, it can add any number of bogies. But what it cannot do, it cannot add the main thing, that is engine. Same thing here also. DNA polymerase cannot initiate the process of replication. But once the starting is done, it can continue. It cannot initiate the process of replication. Why it cannot initiate the process of replication? Because for DNA polymerase to function, it needs, so that is the second point, it needs a free 3' OH. It needs a free 3' OH. So you know 3' OH is found in DNA, 3' OH is found in RNA also. It cannot start replication, but it needs what? It needs a free 3' OH. Since it cannot start replication, we cannot actually have a DNA to provide free 3' OH. So who can provide it? An RNA can only provide it. Okay. So two features we have learned. It cannot initiate replication. Second feature, it needs a free 3' OH for that initiation to happen. And the third feature, it can read the template strand. It can read the template strand only in 3' to 5' direction. It can read the template strand. Template strand means the parental strand. Here this is the template strand. This is another template strand. The parental strand, okay, which is going to replicate. It can read the template strand only from 3' to 5' direction. What it is reading in the template strand? It is reading the base pairs. So according to the base pair in the template strand, it will be synthesizing the complementary new strand. So it can read the template strand only in which direction? Only in 3' to 5' direction it can read the template strand. Okay, and so since it is reading the template strand in 3 to 5 direction, it will be producing the new strand in which direction? In the opposite direction. So the new strand is always produced in 5' to 3'. Okay, so that is the next feature. It can read the template only in 3' to 5', and so it can synthesize new strand. New strand is always synthesized in the 5' to 3' direction. So these are some of the features of DNA polymerase. First, it cannot initiate replication. It needs a free 3' OH. Next thing is it can read the template strand only from 3 to 5, and so it is able to synthesize a new strand only from 5 to 3. So these 4 features you have to keep it in mind. Only then you will understand the process of replication. We have learned the different features of DNA polymerase. Now let's go deep into how this DNA polymerase is doing the process of replication. So as I've told you, always replication is from the open end of the bubble towards the closed side. Here also from the open end of the bubble towards the closed side. So to make you understand, I'm just cutting this bubble into 2. Okay, so when I cut this bubble into 2, I'm getting a structure like this. Like this I'm getting a structure. So this looks like a fork. This is called as a replication fork. So replication bubble is now, I have shown here as a replication fork. And this, the other part is another replication fork. Likewise, in different regions of our DNA, there will be different bubbles, and if you just imagine that bubble into 2 halves, you will be getting different replication fork. And in each of these replication fork, always the DNA replication proceeds from the open end towards the closed end. The direction you should always keep it in your mind. Okay, open end towards the closed end. Now then what happens? Let's see. In this replication fork, when I have cut this into 2, let me make the ends first. So here this is beginning with the 5' end. Okay. Now it is going, and as I make a cut here, it will end up with the 3' end. Isn't it? So the other end will begin with the 5' end. Now how did I make this? Because we have learned in the structure of DNA that the strand is made up of 3' OH is linked to 5' phosphate. So when you cut the strand, what you will get? You will get 2 ends. One end will be 3' OH, another end will be 5' phosphate naturally. So same thing is happening here also. When I have cut this, I am getting a 3' OH on one side and 5' on the other side. Why I wrote here 3 prime? Because this strand is beginning with a 5 prime and so it will end with a 3 prime only, right? Same way, this strand is beginning with a 3 prime and so it will end with a 5 prime. And so the next cut portion will begin with a 3 prime and so it is ending, see here, with a 5 prime. So I am taking now half the portion and I am explaining it. So I can say that this strand is running from 3 prime, 5 prime and so this end will be 3 prime end of this replication fork. Similarly, the other end of the replication fork is going to be 5 prime. Okay. Now let us take the features one by one and let us see. The first feature is it cannot initiate replication. It cannot initiate replication. As I told you, it cannot add that engine to those bobbies. It cannot add. Then who is beginning this process of replication? It is another enzyme. So here let me write the fifth enzyme. It is another enzyme called as primase. This primase is the enzyme which begins the process of DNA replication. Why DNA? Because DNA polymerase cannot begin. So primase is beginning. How this is beginning the process of DNA replication? Primase adds a primer. It adds a primer and then begins the process of replication. So imagine this primer is equivalent to that engine of the train. Okay, so primase is adding a primer. Now what this primer is actually? Okay, so let us see what this primer is. Primer is a short, very short stretch. It is a very short stretch of RNA molecule. Okay, primer is a very short stretch of RNA, or it is an oligonucleotide of RNA, a very short stretch of RNA. Now what is the need of RNA in DNA replication? That is answered by the second feature. This RNA provides a free 3' OH. Okay, because see, DNA polymerase needs a free 3' OH. So that free 3' OH is given by this short stretch of RNA known as the primer. So the short stretch of RNA or the primer, it provides the free 3' OH to begin the process of DNA replication. Okay, so another enzyme, primase, it is adding the primer, and primer is a short stretch of RNA, and that short stretch of RNA is providing the free 3' OH to begin the process of DNA replication. So the first two features or the first two drawbacks, I can say, it cannot initiate, it needs a free 3' OH. These are the drawbacks of DNA polymerase. So that is solved by the enzyme primase by adding a primer. Okay, so here I am putting here, see, here I am adding the primer. This primer is providing a free 3' OH. So here this strand, the strand which is there on the top, the template strand is ending with a 3'. So the newly added strand should begin with a 5', right? So this primer should begin with a 5', and so naturally it will end with a 3'. So that is how it is providing a free 3' OH. So once it got the engine, it got this beginning, and then it got a free 3' OH, then there is no other issues. DNA polymerase can simply polymerize the new DNA based on the complementary sequence. That means if it is adenine here, it will add thymine. If it is guanine here, it will add cytosine. There is no problem. Okay, so DNA polymerase will be keeping on adding new bases which is complementary to this what is there in the parental strand. And for it to add the new bases, what it has to do? It has to first read the parental strand. Isn't it? It has to first read the parental strand. So how it is able to read? It is able to read only from 3' to 5' direction. Okay, so look at this strand, the strand on the top. Replication I told you always from the wider part to the narrow. So here our DNA polymerase can read from 3' to 5'. It can read, and so it can keep on adding. As it is reading, it will be keeping on adding the complementary bases. There is no problem. But the DNA polymerase which is here, it is not able to read from 5'. It is like this. See, when I am telling you a sentence, okay, if I write here, say a sentence, my name is Lakshmi, right? So my name is, I have written. So I can read this only from my left side to the right side. If I start reading from the right to left, it never makes a sense. I cannot read like that. Same is the process happening here. This DNA polymerase can read and understand the bases only from 3 to 5. It can never read the base beginning from 5 to 3. It can never do that. So, the first strand, how it is getting synthesized, it is very simple. But the other strand, the complementary parental strand, how this strand is getting synthesized is a bit complicated. Let's learn about that. Okay, how the other strand is getting synthesized. We have learned like in one strand there is no issues in DNA replication. But in the other strand, our DNA polymerase cannot actually read at all because it is beginning from 5'. Okay, it can read only the strand which is beginning from 3'. It cannot read the strand which is beginning from 5'. But then how this problem is solved? For that, let me just draw this diagram in an elaborate manner and let me just tell you. Okay, see here, instead of structure like this, I am drawing it like this. Okay, this is the closed end and these are the two open ends. These are the two open ends, okay, and DNA polymerase. So this is the 3' end, so this becomes the 5' end. Here this is the 3' end and this is the 5' end. So what I have drawn is a replication fork. I have drawn a replication fork, okay. Now where is my DNA polymerase? DNA polymerase is here, okay, and the part of the DNA polymerase is here also. So always this will be together. This will be together. And always these two will proceed from which direction? From the open end of the fork towards the closed end. It is not like one part will move towards one direction and the other part towards the other direction. It will never happen. Like when a vehicle is moving, its two wheels will be moving in the same direction. No, it is not like one is moving in one direction and the other one is moving backward. Never it happens like that. So same is the case here also. Okay, here also the two structures like this DNA polymerase which are there joined together, they are together called as replisome. Replisome. So DNA polymerase is not a single enzyme actually. It is made up of many subunits. It is a hollow enzyme. Okay, so very big enzyme. So it is like this here. So here when this replication DNA polymerase is here, Primase has added the primer and slowly this area is able to read the DNA template strand and so it is proceeding towards this direction. No problem at all. But here this DNA polymerase is not able to read at all. So it cannot proceed. So then what happens is it starts reading from this direction. So if this is 5' end, this end of the template will be 3' end. This DNA polymerase which is present here, it starts reading from the 3' end towards the 5' end. So it reads like this. It has to go forward but it reads backward. Okay, because it doesn't know to read from 5 to 3. It can read only from 3 to 5. So it has to go forward but it is reading backward. So when it is reading backward, the primer has to be attached to this end. towards the 3' end of the template strand. Like here, this is the primer and this primer is attached to the 3' end of the template strand. Same way, in the other strand also, primer is attached towards the 3' end of the template strand. So when primer is attached to the 3' end, the primer will begin with which end? 5', the opposite. And so the primer will end with which end? It will end with 3'. Isn't it? So what is getting formed here? What the primer is actually giving? The primer is giving a 3' OH. And now things are it is quite okay. Now DNA polymerase can polymerize from this end towards the other end. But here see, once it polymerizes from here to the other end, it cannot further proceed. But on the top, if you see, it can keep on proceeding like this. This process can keep on going. As this replisome is moving forward and forward, new and new base pairs will be added here continuously. But here, as the replisome is moving forward, it cannot add like that. Why? See, it has added till here. Now it has to start anew from the other area. So that is, see, if this replisome has moved one step forward, I am drawing like this. It has moved one step forward here. This replisome is now here. It has moved towards this side. Okay. So as it is moving here there is no harm. It keeps on replicating. It keeps on adding the bases which is complementary to the parental strand, because it can read from 3' to 5'. But the other strand, it cannot read from 5 to 3. So it is reading from 3 to 5. So when the replisome is moving one step forward, it will again start reading from 3 to 5 of this strand. Okay, so it is again starting as a new. So when it is again starting as a new, again it needs a primer. Again it needs a primer. And then it will synthesize like this. Okay, see, it is synthesizing like this. Okay, and there is a gap between the first one that is synthesized here and the second one. This gap is there. It is filled later on. Again, when this replisome is moving again one step forward like this, again when it is moving like this process is continuously synthesized. But here again a new primer has to be formed, and then the synthesis direction will be like this. And again there will be a gap. So if you just compare the replication in both the strands, it is entirely different. In this strand, in the parental strand, which is running from 3' to 5', since DNA polymerase can read this template strand in 3' to 5', the new strand is continuously synthesized here. And so you call this strand which is continuously synthesized as the leading strand. Okay, so this leading strand will be complementary to the parental strand which is running from 3 to 5. Because our DNA polymerase can simply read from 3 to 5. And since it can read from 3 to 5, it is always synthesizing the new strand from 5 to 3 direction. So that continuously it is doing. No problem. And this leading strand needs only one primer. One primer. And there is no gap here found. So those are the features of leading strand. It is continuously synthesized. It is always synthesized to the template which is running from 3 to 5. It needs only one primer and there is no gap in between. Now what about the other strand? This other strand is called as the lagging strand. Lagging strand. Because replisome is moving towards one direction, but it is formed in the other direction. So once a little bit of it is formed, again it has to start anew, again it has to start anew. So every time that it is starting anew, it needs primer. It needs primer. And it is not continuous. It is produced in small fragments. And these fragments, these small fragments are called as Okazaki fragments. Okazaki fragments. after the name of the scientist who solved the mystery of how the other strand is getting replicated. Okay, Okazaki fragments. The fragments which are synthesized, or the fragments which are synthesized complementary to the parental strand that is running from 5 to 3. So this strand is always synthesized as fragments, and this is called as lagging strand because every time it has to start anew, okay, and it needs 1 primer every time that it is starting, and there are gaps in between also. So this is how the other strand is getting synthesized. And so as this new strand is getting synthesized, this will become double-stranded DNA. This also is going to become double-stranded DNA, and helicases will now unwind these hydrogen bonds here. This bubble will be keeping on expanding as I have shown you in the previous figure. It will be keeping on expanding and new DNA strands will be formed. So leading strand and lagging strand. Now the question is how these gaps are getting sealed. How these gaps are getting sealed. And how this RNA is getting removed. This is actually RNA. The primer is RNA, isn't it? Short stretch of RNA is primer. We don't need RNA in the DNA replication process. So those primers have to be removed and then these gaps have to be sealed. Now these two processes are left. That is also done by DNA polymerase, but not this DNA polymerase. So what is that? Not this DNA but some other DNA polymerase. So let me tell you, DNA polymerase, as I have told you, is not a simple enzyme, but it is a complex enzyme. And DNA polymerase is not just one enzyme. There are 3 DNA polymerases. DNA polymerase 1, 2 and 3. So whatever feature I have listed here, and the real hero of DNA replication is actually DNA polymerase 3. Okay. Now this DNA polymerase 1 is doing the function of removing the primer. It removes the primer. Okay. It removes the primer and then adds new DNA in its place. It removes the primer and adds new DNA in its place complementary to the parental strand. And this RNA polymerase 2 is involved in proofreading. It goes and checks whether the replication process that is going on is taking place in a proper manner or not. That means whether if it is adenine in the parental strand, whether thymine is added or not, or if something else is getting added. So it is going and reading the proof whether things are properly done or not. So see how miraculous things are happening. So DNA polymerase is not just a single enzyme, there are 3 enzymes functioning. DNA polymerase 3 is the main hero who is having all these features and doing this process. Then comes the role of DNA polymerase 1 and 2, which is coming and clearing out all the things, and then they are proofreading. So here I told you, removing the primer, proofreading. Now one more thing is left. What is that? Sealing this gap is left. Isn't it? Primers are removed, proofreading is done, and we have to remove the gap. Remove the gap means what is the gap here? If this is the 3' end, here this will be again the 3' end, this is the 5' end. So there is a gap between the 5' phosphate and 3' OH. This gap has to be filled. That means what bond has to be formed between 3' OH and 5' phosphate? It is a phosphodiester bond. So there comes the role of the last enzyme, the 6th enzyme here, that is ligase. Ligase is the enzyme which seals this gap by forming the phosphodiester bond. So the gap is sealed, DNA synthesis is completed here. I hope you have understood. So let's just summarize from the beginning the whole process of DNA replication. First, the 2 strands of DNA get separated at different origins of replication. Okay, so when they get unwound at different origin of replication, it is done by the enzyme called as helicases, and these helicases breaks open all the hydrogen bonds one by one and the two strands are separated. Now, the enzyme Topoisomerases and single strand binding protein helps in keeping the two strands separated till the process of DNA replication is over. As and how this replisome is moving forward, whatever clips was here, no, single strand binding proteins, I told you it is holding the two strands with two clips on either side. So as the DNA polymerase is proceeding, the two clips will be removed and removed. Single strand binding proteins will move away from that place. Okay, and this replisome will be proceeding to add new and new DNA molecules. So here the main problem is that our DNA polymerase cannot initiate. So that is initiated by the enzyme primase by adding primer. Primer is a very short stretch of RNA which provides the free 3' OH. So when the free 3' OH is available, our DNA polymerase can continuously synthesize it. It can continuously synthesize the strand only to the parental strand which is running from 3 to 5. Because it knows only to read the strand that is running from 3 to 5. It cannot read the other strand because it is starting from 5'. So what does it do? It starts reading from the end. Where it is, from that end it starts reading towards the other side. So what happens is it cannot synthesize continuously. So that strand becomes the lagging strand. And every time it is beginning as a new, it needs new and new primer. So at the end, when the process is over, what is done is these primers are removed, proofreading is done and the gaps are sealed by the enzyme ligase. So there ends the wonderful process of DNA replication. Hope you have understood the whole mechanism that I have taught, okay? And if you have any doubts, please feel free to post it in this channel, your doubts, your comments. I'm waiting for all those things. And if you haven't subscribed the channel, please subscribe it. And in the coming videos, I will be posting one very important feature of DNA replication. That is, DNA replication is said to be semi-conservative. So semi-conservative because always when this DNA is getting replicated, always in the newly formed DNA, one strand will be parental and one strand will be new. Okay, so when one DNA is replicating into two, what is happening is always in the newly formed DNA, one strand will be always parental strand and the other strand is new strand. That new strand is produced by DNA polymerase. So always one parental strand will be there. That is why it is semi-conservative. So in the next video, I will tell you the experiment which proved the semi-conservative nature of DNA replication. So till then, thank you all of you.